to the signal detection webinar. So just wanted to really start with the legislation and guidelines, and certainly with the new EU legislation, this has been a focus for implementing the new legislation. And we can see that before there was obviously always a requirement to perform signal detection. However, it was realized that there needed to be a bit more focus around this, a bit more process around this, and justification for decisions that are made. So the relevant legislation, first of all, we have the implementing regulation, which came out around the time of the new EU PV legislation and provided a lot of information with regards to how signal detection should be implemented according to this new legislation. We had Module 9, which was also referring to signal management, EU specific, and then we had the Q&A on signal management, which was last updated in September 2014. However, this was probably more relevant for the regulatory authorities and use of actitivies rather than practical aspects for pharmaceutical companies as a whole. Then we have our global publication, CEOMS 8, which was really about what types of mechanisms you can use for signal detection, statistical analyses, etc., and the pros and cons of using these. And I put ICH E2C here. This is related to PBRAs, and that's again because with the new legislation there is a focus on ensuring that you have detailed information presented in the PBRA with regards to your signals overall. So let's look at the definition of a signal. And it really, I mean, the way we think about it, it's a signal that something's not quite right with what you're seeing. But this is a definition that comes from CEOMS 8. And you can see they talk about signal arising from one or more multiple sources, suggesting a new actually causal association or a new aspect of a known association between an intervention and an event or set of related events. And they do put here either adverse or beneficial and sufficient likelihood to need some kind of action carried out. And it's interesting that we have adverse or beneficial here. Normally we do think of signals more on the adverse perspective rather than the beneficial. Now, I just wanted to look at the overall process as a whole, and this is well documented within GVP Module 9. You look at your signal detection activities, your validation, prioritization, assessment, and then recommendations for action. You need to consider exchange of information all the way through this process, through these different steps. And it shouldn't be something that's just done at the end, because it may be that as you're going through the steps, you realize that you have either an urgent safety issue or an emerging safety issue that does need communication in some way, either to the health authority or to the health authority first and then to healthcare professionals. So you do have to bear that in mind the whole time as you're following the signal management process. And I think it's worthwhile pointing out that whilst we talk about signal detection, and a lot of people talk about signal detection, really detection's one piece of it, and it's a signal management process as a whole. So now focusing on the first bit, the signal detection side. So this is where you're looking for something that's not quite right in the sea of data that you have. So apologies. Sea of data that you have. So you need to ensure that you consider all of your data sources, not just your cases, but safety information, alerts can come from anywhere. So for example, you have quality here. If you have a manufacturing issue, that could be safety related. That could have implications for safety, and you would need to ensure that you're aware of anything that could have that implication. Clinical, we're quite used to having those conversations, understanding what safety issues may arise from clinical data. Non-clinical, you could have a animal study that shows some kind of anomaly, and you may need to consider whether that's relevant for the human population or not. PV activities that are ongoing, it may be that something comes out from a post-authorization safety study that you need to further consider, pharmacoepidemiological data, and the literature. And certainly for your older products, if you will find a lot will come from the literature. It may be that somebody does a study looking at a specific issue with an older product, and suddenly you will see that there's a potential for an association with a specific issue. So, for example, recently we had the benzodiazepines and dementia that came up as a study that was performed and was looked at the PRAC level. So moving on now, looking at the different methods overall, these can be divided up into kind of the review of your individual cases, your ICSRs, looking at your aggregate data overall, and data mining. And we'll come on to each of these over the next couple of slides. So looking at your traditional methods, and these tended to be more of your qualitative signal detection and focused around reviewing your cases individually. It could be a single case was seen with Stevens-Johnson syndrome. That was enough for you to actually investigate further, because Stevens-Johnson syndrome is normally associated with medications more than some of the other events. If you have a single case, that might be sufficient for it to be an actual alert that you need to then perform the validation, assessment, etc. Also, you could have your line listings as well, and often a lot of companies will have, for example, your serious, unexpected, related listings, and it'll give you an idea of what's occurred over a certain time period, and you would review them to see if any pattern of abnormality is being seen. You can look at your aggregate data, as you would do for your PBRAs, etc. And whilst you're not meant to use your PBRA for signal detection, it is a different set of data. Well, not a different. It's a different way of presenting the data that might again show you that there's an issue that you need to look at overall. Absolute case counts, where you're seeing an increase in the number of cases. So you could see 10 cases of event X, which suddenly means that actually you're a little bit concerned that you're seeing this number of those cases. That could also be something that flags to you that there's an issue ongoing here. Or you could look at simple reporting rates, where there's an increase in frequency, or where there's a exposure-adjusted reporting rates, taking into account the exposure of the product overall. But all of these are very much, based on, qualitative analyses. Then you don't have statistical methods behind them. It's about eyeballing the data, looking at the data, going through it, reviewing it, and picking out what you think is abnormal. Looking at the data mining tools, now these are your statistical methods, and they're much more sophisticated, and they're all focused around having something that's disproportional from a drug event perspective. So often you will find that a company will have their traditional methods as well as adding on the data mining tools as well to make sure they don't miss anything. And you need something like this when you have a large number of cases, because you can't physically go through everything. You need some way of being able to trawl through the information that you have and being able to pick out something that's not quite right. And what you're doing here is really looking at disproportionality. You're looking at whether what you're seeing with your drug is in accordance with what else you've seen with your drug. So you could look at a PT relevant to all the other PTs that are there on your database with your product, or in line with your database as a whole, considering all of the products that you have. Is what you're seeing with your drug, out of sync, disproportional to what you're seeing with all of your other products as a whole. You do need to, though, understand the limitations of your data, where that information has come from, the structure, how it's been put into the database, and exactly what is captured on that database. Now, looking at the databases overall, there are many that are available. We have AIRS, FDA, sentinel, which I think has been changed. That was the MHRA one. Vigilance as well. Obviously, huge databases, lots and lots of events on them. And you also have a dedicated vaccine database for vaccine products. But you do, like I said, you do need to know what you're looking at and what you're using as baseline, because the older databases may have products that aren't available anymore, and this will skew then what you're using as your baseline. So looking now at the different types of statistical methods. So I've detailed them here. You have your PRR, which is your proportional reporting rate, and here you're looking for where what you're observing is more than the expected value. And with these statistical methods, you'll see an observed rate against the expected value, and there'll be a statistical analysis to say, right, you know, there's a probability associated with seeing that event, and if the p-value is less than a certain amount, then we could say, well, actually this is unlikely to have occurred by chance, and there is an alert here, and that is what you're doing with these statistical methods. So you look at overall your number of AEs, your absolute number, and you compare, like we said before, either to the number of reports you have for that product, or for the number of reports that you have on the database as a whole, or for a class maybe of products, you know, so you're comparing what you're seeing with your product to the class of products that are on your database. The problem with PRR is that you do get a lot of false positives, which means then you will spend a lot of time looking at alerts which are spurious and don't need looking into. So there have been other methods that have been introduced to try and reduce this. And one of them is, well, these are called the Bayesian methods, where they are looking to reduce your false positives. And there are a couple of formulas that are used with regards to this, but all based on the same premise that you're looking for disproportionality overall and then statistical significance associated with that. There are a few slides at the end, 3 slides, which talk about the PRR and the Bayesian methods in a bit more detail should you need. But I wasn't going to cover that in this webinar specifically. What you can, though, do is use the PRR and the Bayesian methods as well, and you can plot your PRR values, your Bayesian outputs, and have a look at trending that way and see exactly what is going on with your product. So you could present them graphically. So in summary, for signal detection, you do need to ensure that you have the appropriate method for the data set. I mean, there's no point having complex stats if you've got like 2 cases that occur per month. You're not going to add anything with that. You need your traditional quantitative measures, and that's enough. That's all that you would need for that. You need to consider data from all sources and make sure that you have a process that covers that, so everything comes into you for review. Your curative outputs also need to be assessed appropriately by a qualified person in a timely manner, and that was a key part actually of the legislation, of the EU legislation, that you need to have someone that's adequately trained, that knows what they're doing. And you need to ensure that you can document that process and you have a rationale for the method that you use for your product and the periodicity. And it's justifiable. So if you have a product that's been on the marketplace for many years, not many events coming through, it may be appropriate. It's appropriate to look at that once a year, for example. However, if you have something that has high usage and it's new to the marketplace, waiting a year is not going to be appropriate. It's worthwhile noting that in the future, vigilance will be available to MAH. It is available now, but it will be more readily available, and MAHs will be expected to assess or interrogate vigilance for signals as well. The EMA do this already, and that's where you will see the PRAC signals coming from. And interestingly, what they have shown is that the signals that come from the vigilance database are very often ones that aren't picked up by the MAH because it's a different data set. I just wanted to show this because I think it's useful to have a look and see what you need to have in place to be able to detect various signals or various alerts or pre-signals. So if you have something that's common as far as in the patients taking the drug, but rare in the background, So here we've got an example of phocomelia due to thalidomide. You will pick that up quite easily through clinical observation. If you have something that's a bit rarer in regards to the product and rare in regards to background as well, you'll probably still be able to pick that up on clinical observation, but it will be less easy. It will take probably longer, more reports to come in. And here we have the example of aspirin and Rey syndrome. Then if you have something that's common in the background and common with the product, you're probably going to need a large kind of study to be able to pick that out. And certainly if we look at ACE inhibitors and cough, that was only picked up after a large study was performed. And then if you have something that's uncommon in the drug, patients taking the drug, but common to rare in the background, you're going to need an even larger study to be able to pick that out. And here we've got an example of HRT in breast cancer. And then if you have something that's rare with the drug but common in the background, that's going to be almost impossible to pick out, and we don't actually have any examples of that. But I think you can see from here that depending on what type of alert that you're seeing and the relationship to the product and the background rates, etc., you are going to need lots of different mechanisms for being able to pick these things up. So now just wanted to talk about signal validation, and there's been a lot of discussion about validation and exactly what it means, because this was a step that was really introduced by the new legislation. And really what you're trying to do here is say, right, we've got an alert, but not all alerts are going to be signals that need assessment. You need that interim step, and that's what this interim step is to say, right, do we have enough supporting evidence to warrant then a full assessment of this alert? And you need to take various things into consideration when deciding whether you're going to validate it, well, whether a signal is validated or not. The clinical relevance, the novelty of the reaction, the previous awareness, because usually these are new things that have come about, and looking at information around that whole alert. So you could do a top-line literature review, look at whether there's a biological mechanism that's plausible, looking at databases and whether it's seen as a class effect, looking at your D-challenge, re-challenge, temporal association. You know, what evidence do you actually have with regards to that alert and the causal relationship with your product? And it's that validation step that is a little bit new, but does have to be documented. And, you know, we'll come on to that when we talk about tracking as a whole. It has caused a bit of problem for people, because I think people find it hard to go from an alert to a validation step and then to assessment. I think people are more used to going straight from an alert to assessment, but there is this step in between, and you have to really say, right, is what I'm looking at something that I really do need to look at further or not? And that's what you're doing here. So if you do have a validated signal, you now need to prioritize, and you need to prioritize because you can't really assess everything all at once. And for prioritization, there are various things you should take into account. If there's something obviously that's extremely urgent, you would want to prioritize that above the others. But you need to look at the impact on the patients overall, whether something's severe, whether it's reversible or not, whether you can prevent it, the clinical outcome. All of these things will help you decide what you should be focusing on initially. It very much follows actually CIOM's for the benefit risk assessment and kind of the decision process in that's detailed, looking at your product, what it's used for, looking at the actual issue and the seriousness of the issue, severity, reversibility, whether there are other treatments available or not, the strength and consistency of the evidence that you've got, the impact, public health impact, whether it's already known and this is an increased frequency, or whether your product's only just been on the marketplace and this is something very new that needs to go out there very quickly. They do actually state in Module 9 that certainly when you have something that has a potential for high media coverage, you should consider prioritizing that as well. I think that's a good point because, you know, not only we are looking obviously at things from a safety perspective, but these are the things that do create a huge problem for us as an industry, and does impact from a safety perspective, because all you need is a huge urgent issue out there that's blown out of proportion completely, and you have to deal with the fallout from that. Right, so now coming on to the signal analysis side. So here, most companies do have their own template for how a report should be done. Usually it's a report when you do your signal analysis. And you need to look at what sources you're going to review, what's available to you. And again, it will depend on the type of product that you have. If it's an older product, you may well not have access to the clinical data. You may not have access to much of the preclinical data as well, because things have changed so much. If it's a newer product, you might have more clinical data than you do on your safety database. So, you know, you need to kind of understand where your product is overall within its life cycle. You also look at preclinical data. Was there anything there that would point towards this being an issue in the human population? Epidemiology obviously is useful, give you the background rates of what's going on, so you can look to see with regards to the issue that you're looking at whether this is over and above what you would expect. Normally you would do a literature search, you'd think about the product and the issue, but also look at the class as well and the issue. Textbooks, of course, and I think importantly you do need to have multidisciplinary input. It's not just the safety representative that should be doing this completely on their own. You need to have input from all of these different areas to make sure that you've covered all your bases. And you do have to have a clear strategy of what you're trying to answer, because very often you can start to write a report and then you can get lost completely in the data and start answering the wrong question. And it is extremely important to make sure that you know what you're answering so that your strategy is appropriate. So looking now at conclusion overall. So conclusion for your signal is that refuted, no further action is needed. This signal, there's no issue here, nothing else to be done. Or you can have an identified risk, which can be important or non-important. So this is something that's related to your product and you will need a label update. Or you can have a potential risk, which is either important or not important. So there's something going on, but you don't have enough evidence as yet to be able to say that there is a causal association in some way. So those are your three things that you can conclude. And the reason that we put it, or I put it like this, is because within the PBRA, this is what you're meant to present. You're meant to present, if a signal is closed, what has been the conclusion in this way, and then you're meant to cross-refer to the relevant sections within the PBRA. As in, if it's an identified risk, you'd put it into the identified risk piece. So you have your conclusion, but then you will also have your recommendations for action. So even though you might have an important identified risk, you may need to actually put additional risk minimization into place. So this is where, right, we've concluded, now what do we need to do? Do we need to do anything else? And again, you need to consider this throughout the process, not just at the end, because you might have an emerging safety issue or an urgent safety issue that needs dealing with immediately. You might need to look at suspending your marketing authorization. I mean, that's quite a severe thing to do, but it has happened many a time. It may be decided that you need more information, so maybe you need to put something else into place. Additional pharmacovigilance activities may be required. It could be that you decide that actually we're just going to review this signal periodically through the PSURs. Maybe it's now an important potential risk, which will mean that it will be reviewed regularly within your PBRA document. You might need risk minimization activities. So say if there is an important identified risk and you decide that you need some educational materials, that would need to be discussed here. Update to the product info. If it's an identified risk, important or non-important, that means you know that there's a causal association and you're going to update your product info. You're going to add it to the label. Could be that you need to do a post-authorization safety study to exactly work out what's going on. You've got a potential risk and you need further information. But in each of these situations, you need to specify the reason why the action is appropriate and the time frame for completion. You need to ensure that appropriate progress reports are produced and that they are sent to the relevant place and reviewed appropriately as well, depending on the severity and impact on public health of that particular issue. So looking at exchange of information, we've talked about that and the fact that you need to assess this all the way through your signal management process. But you need to ensure that there is an appropriate time frame for exchange of information. So if you're going to update your label, you need to ensure that you have a time frame that is appropriate for that update. I know in certain companies and in certain health authorities as well, they use a 1 month, 3 months, 6 months to give you kind of an idea that if it's urgent, 1 month update to your label, and there's discussion then whether that's in packs that are out in the marketplace or whether that's just update at the wholesalers, and that does actually differ company to company. 3 months is for a medium issue, and 6 months is more for a routine, not a routine update, but a non-urgent signal, I should say. And then you would need to submit that to the health authorities, and then you would eventually have that go out to the healthcare professional through your updated label and your patient information leaflet. Now, if there is a significant or urgent safety issue or something that impacts warnings and precautions, for example, you may well need a direct healthcare professional communication as well. Now, throughout your whole process, if there's an emerging safety issue, you need to ensure there's prompt notification. This is covered actually in Module 6, where they talk about emerging safety issues and what constitutes an emerging safety issue. And I would suggest going back there to look through if you're not sure. And then, of course, you have PBRA, which details all the signals that have been reviewed and the outcome of that signal analysis during that time frame. Now, tracking is a big thing from the new legislation, and the fact that everything needs to be documented down. Your justification, for your different activities, and seeing when you've done exactly what part of that signal management process. So for validation, you need to track what you used actually for your validation. So if it was just based on case analysis, and then say if that event or that issue comes up again, somebody can go back and say, right, actually this wasn't valid at that point, but now we've got further information and we think it is a validated signal now, and it will help overall in that process. So you need to state when it was identified, what your validation step was, and when you validated it. If it was not valid, why not? If it was valid, justification for the prioritization. Overall, your assessment, what was the outcome and the rationale for that, and then timelines for any activities that are associated with that assessment. So whether that be risk minimization activities, a study, or updating of your label. And then really getting that information out to the health authorities, healthcare professionals, patients, etc. So all of those key steps need to be tracked appropriately with justification for why those decisions were made, and it needs to be archived. You need to have this audit trail available in an inspection setting. So looking at the strategy overall, I think if you're looking at putting a strategy into the organization, you need to understand that no one size fits all. It depends on the type of products that you have, the data that's available, but you do need to consider all of those elements that we've talked about previously. And it may well be that statistical analyses and data mining is not appropriate, which is fine, but there needs to be that justified decision as to why that's the case. I just wanted to touch the last two slides. One is on quality. So looking at quality, again, very much focused on within the new EU legislation. You need to have people with the appropriate expertise to perform these tasks, and they need to be trained, and this needs to be documented so that you ensure that you have the right people performing these activities that are able to make the right decisions. Roles and responsibilities need to be very clear, and whilst we have this whole thing about cases and cases being reported, people in other disciplines need to understand that if there is a potential signal, that they need to forward that to the relevant person so that it can be documented and reviewed appropriately through the whole process. QA and QC obviously goes without saying for the whole process, and an audit trail, as we've just talked about, and also for the queries that come in and outcomes, so really end to end. Now lastly, I just thought it's worthwhile mentioning that in an inspection setting, these are the things that will be looked at, and I think it's worthwhile pointing out that signal detection, management, following through on label changes or label updates based on signal management are a big issue for inspections, especially for the MHRA, and they're one of the main reasons for critical findings in the last few years. And it really is that end-to-end process of your alert right through to updating your label. And there are deficiencies in timeframes and process as well, but a lot of it is timeframes where things don't end up getting into the label, your SPC, or other labels within an appropriate time frame. So you've identified something, you said there's an issue, and you haven't updated your label. So really, they're going to look to make sure that you've looked at every source available, appropriate source available, that you've appropriately applied your methodology for analysis, your investigations are appropriate, and follow-up actions that are suggested are appropriate as well, and that you have actually updated your label as you said you would do, and that communication to external to the external audience is appropriate. So I think I'm going to end it there. That's a very quick kind of run through signal detection. There are additional statistics slides. I will just click on them so that you can stop and have a look at them. This just shows you the PRR. This is where you look at a table, looking at what you've got for your drug, looking at other drugs, looking at other events, and then you can get a proportional reporting ratio. And then looking at your Bayesian methods, they're detailed here as well. I'm not going to go into a huge lot of detail, but there is a very detailed assessment within CEOMS 8 that discusses how these methodologies work. And if you're interested, you can go and look in CEOMS 8. Very often what you'll find is a value and a statistical significance, and that's enough for you to say, yeah, this is an alert I need to look at. Okay, I'm going to stop there. Thank you very much, and apologies for the issues that we had with the recording before. Thank you.